Hi everyone. Let's begin with the next topic of general microbiology, and that is bacterial anatomy and physiology. Under bacterial anatomy, this is going to be a topic not only important for the second prof exam students, but every word and every sentence of this is important for also your MCQs and how these concepts would be integrated eventually with the bacteriology section. So the first layer that I'm going to start with happens to be the outermost, known as either a capsule or a slime layer. Now, firstly, what is the difference between them? Because either bacteria will have a capsule or a slime layer. Both of them can't be there. So what is the difference between these two? Capsule is something which is very tough. It is very well demarcated. It is very rigid. Whereas slime layer is very loose, or you can other words say it is undemarcated. In other words, it is undemarcated. I'll show you what slime is. I hope all of you have seen this over the internet. A lot of toys of kids are made from this slime. It's very soft, it's very stretchy, jelly type. And that is what slime is. Slime is very, very loose and undemarcated, and capsule is extremely tough and demarcated. So like I said, either a capsule or a slime layer would be present, because is there any organism which has both? Yes, when you go to bacteriology, I have taught you that Streptococcus salivarius. Streptococcus salivarius has both a capsule as well as a slime layer. Both the things are present, the only one to have that. But why has God given a capsule? Now you know that if a bacteria, go back to your pathology lectures, if you know a bacteria comes into your body, if this is a bacteria, what are you going to coat it with? You're going to coat it with some kind of opsonin, remember IgG and opsonin, so that you can eat it. But this capsule will say that I will not let opsonization occur. I will prevent this opsonization. As a result, you will not be able to eat the bacteria. Purpose solved. God has given the capsule basically so that the bacteria is protected. It's like a shield around the bacteria so that you cannot eat it up. Now you will ask me, what are all the capsulated organisms in the body? And I have that smile on my face because I know it's not an easy mnemonic. I've tried my best to assimilate things. Pretty nice capsules. I don't think they're pretty nice because the mnemonic's not so nice. But please note, pretty tells me it's all the P's that you can think of. So if you ask me which Streptococcus, I'll say Streptococcus pneumoniae. Even a little bit about Streptococcus pyogenes I'll tell you. So Streptococcus pneumoniae. If you ask me, okay, Klebsiella which species? Klebsiella pneumoniae. Bordetella has many species, but Bordetella P wala, pertussis. Vibrio has many species like Vibrio cholera, Vibrio vulnificus, but I'm interested in the P, so Vibrio parahemolyticus. Clostridium has a lot of species like Clostridium difficile and all, but I'm interested in the P, so Clostridium perfringens. Yersinia has a lot of species. I'm interested in P, that is Yersinia pestis. So I realized that whatever the examiner will write in front of me, I will pick up the P's of that member. Streptococcus pneumoniae, Klebsiella pneumoniae, Bordetella pertussis, Vibrio parahemolyticus, Clostridium perfringens, Yersinia pestis. All the P's, that is what we call as pretty. Now when you say nice, nice sounds like Neisseria. Nice sounds like Neisseria. Which Neisseria? Meningitis or gonorrhea? Meningitis. Let me tell you the structure of meningitis. The way you write an M, from the M you can make a lens. It looks like a lens, and around the lens there is actually a capsule. So Neisseria meningitis is a lens-shaped capsulated organism. Not only is the N for the nice, I is for influenza. We know there's only one very famous Haemophilus influenza. Pretty nice capsule. So C, we also include a fungus. That fungus is Cryptococcus. And A is going to be the most important of this family, that is Bacillus anthracis. I'll tell you the importance, but also one tiny little one, that is A for aureus, Staph aureus. Now what is it? Pretty nice capsules. May please note that all the capsules, they are made up of polysaccharides. All the capsules are made up of polysaccharides. However, I can see some exceptions. I can see Yersinia pestis where a peptide is written, and I can see Bacillus anthracis where a peptide is written. So these two exceptions need to be learnt. Very, very important exceptions, Yersinia pestis and Bacillus anthracis, in which we have a peptide wala capsule, not a polysaccharide. It is a polypeptide. Next, what you need to know is there is an organism called Staphylococcus aureus which has a very tiny capsule, might not even be visible. So that is known as Staph aureus, which is another exception, which has a micro capsule for you to know. Another thing that I'm going to tell you, which is not in this list, please note Streptococcus pyogenes at times can also have a capsule, and that is made up of hyaluronic acid. That is also not a polysaccharide. That is hyaluronic acid, which is important. So that makes up all the capsular changes. These are bullet points. You know, it does the examiner like rules? No, examiner loves exceptions. Quickly repeat. Yersinia pestis and Bacillus anthracis are polypeptide. Staph aureus has very small microcapsule, and Streptococcus pyogenes at times can have a hyaluronic acid capsule. So now when you're talking about this, how do I demonstrate? There are two very important tests in microbiology that you study, Mcfadyen and Quellung, and I'll give you mnemonics for it. For example, you want to do Mcfadyen's reaction for a capsule called Bacillus anthracis capsule. What will you do? You will take the bacteria and you will add a stain called, you'll say what stain? So think Mcfadyen mein you will add M wala stain. You will add something called polychrome methylene blue. You will be adding something called polychrome methylene blue, because blue color means I'm going to get something blue out of it. It's going to give a purple color around the capsule. There's a purple color obtained. So please note, for this is the question that you get. What is the stain used for Mcfadyen's reaction? We use the polychrome methylene blue. Next, we have a testing called Quellung reaction or Neufeld reaction. Like for example, we are doing it for Streptococcus pneumoniae. You can do these for any capsulated organism. I'm giving you most important examples. So Quellung, I have rhymed it with swellung or swelling. Quellung will remind you of swelling. I'll tell you how. For example, if this is a bacteria and all of you are saying that I'm talking about capsulated bacteria, so can I say that every capsule will have an antigen around it? And if there's going to be an antigen, I think this much everyone knows that if I add an anti-sera or an antibody to it, there will be a reaction. There's an antigen-antibody reaction that is definitely going to take up over here. Will that result in the swelling of this capsule? This capsule will end up swelling. That is important. So please note that when they talk about swelling of the capsule, they are talking about which reaction? Swelling for Quellung. Easy. And I also call it the Neufeld. In how I have learnt it is fela hua capsule. Okay, so Quellung is gonna be the swelling of the capsule, and the Neufeld means fela hua bigger capsule. These are the two tests, Mcfadyen and Quellung. But what exactly is going to be the modification of slime known as? If this gets modified, now some of the bacteria will have it. That is going to result in a biofilm. I'll give you an example. You must have heard that there is an organism called Streptococcus mutans. In case you haven't heard, let me tell you. Mutans goes in mouth. Mutans, mouth. Yeah, what does it cause in the mouth? It causes dental caries. Means it's going to cause disruption to your teeth. It's going to cause damage to your teeth. Why? Because it forms a film on your teeth. Which film? It forms a biofilm on the teeth. That is why it causes dental caries. Similarly, you will study there is an organism called Staph epidermidis. Staph epidermidis, and you will study this in the staphylococcal chapter. This is a prosthetic heart valve endocarditis. That is what it causes. That also results in the formation of a biofilm. So I am seeing that it's all the danger things are happening. Dental caries, valve endocarditis. That means this biofilm is no simple person. Biofilm is quite deadly. So what is this biofilm going to do? Number one is going to help in sticking. The bacteria is going to stick to that place, adherence. Number two, if I will try to go and eat it, phagocytosis, it will not let me eat it. It is antiphagocytic. Number three, if I give any antibiotic to kill it, it will not let the antibiotic also kill it. So antibiotic resistance. So please note everyone that number one, it is going to help in attachment. Number two, you will not be allowed to kill it. That is antiphagocytic. You will not even have the antibiotics kill it. Antibiotic resistance. And that is what you have for a biofilm. Please note that biofilm can also be shown by Pseudomonas aeruginosa. Pseudomonas aeruginosa can also show a biofilm. All these examples you will be doing. So the first layer for all of you is done. You're done with the capsule. Now you're going to move on to the next layer that you have, the very famous, in which you get maximum questions, cell wall. Let's get going. First, I have to differentiate for you guys between the cell wall of a gram-positive organism and a gram-negative organism. And I'll give you some mnemonics. To begin with, your mnemonic is PPT. To begin with, I'm just saying PPPT. Why? Because I'm talking about gram-positive organisms. And if I say who has more of peptidoglycan, you will say, of course, gram positive has maximum peptidoglycan, so much so that it has become 100 layer thick. Whereas gram negative, see, peptidoglycan is very less. It is very less. Primarily, it is made up of lipopolysaccharides. So see, peptidoglycan is very less. How less? Hardly 1 to 2 layer thickness. Let me show you a picture. Have a look at this image. I hope everyone can see that the peptidoglycan layer is so thick over here. So this must be a gram positive organism. And the blue color peptidoglycan layer is so thin over here. So this must be a gram negative organism. So P for P. If the peptidoglycan is more, positive hai. If peptidoglycan is not there, negative hai. So what does the gram negative contain? Gram negative contains a huge layer over here called lipopolysaccharides. That is what it has compensated. So now tell me, if I'm saying gram negative has lipopolysaccharides and I ask you who has more of lipid content, common sense again, that gram negative will have a lot of lipid content and gram positive will have less of lipid content because lipopolysaccharides are more in gram negative. Sorted? Let's move forward. What else do I see in a gram positive cell wall? I see that there is some thing known as this teichoic acid. So firstly they will ask you where is a teichoic acid present. Teichoic acid is going to be present in a gram positive organism. So abhi tak what is my mnemonic? If I'm dealing with a positive organism over here, then I told you no PPPT. So I have done that. Number one, P is going to be peptidoglycan, which I have seen. Next, now I'm telling you that a teichoic acid is also going to be present. So the T is done. So you'll ask me the function of a teichoic acid. Can you learn, you know, the Hindi word teichoic sounds like tikna. If you have to kahin tikna hai, means if you have to attach somewhere, that is what you will use a teichoic acid for. So if they ask you teichoic acid is going to be present where, you will say teichoic acid is present in gram positive organism and it is absent in a gram negative organism, and teichoic acid is mainly for attachment purposes. That is the purpose of a teichoic acid. Okay, so I realize more or less it's coming to an end, but is there something that only a gram negative has? Because my most focus has been on gram positive. Gram negative ke paas to kuch hai hi nahi. I am saying peptidoglycan is also not there, teichoic acid is also not there, to hai kya? If I see carefully, God has compensated. Firstly, God gave it a lot of lipid, lipopolysaccharides. Second, God has given couple of extra things to it. If you look at this guys over here, you can see a very, very famous periplasmic space, an extra space. You can see another membrane because peptidoglycan was not there. Now you're trying to compensate. God has given it another membrane called outer membrane, and then God has given it some pores called porins. So what are the extra things that I'm seeing? I'm seeing the periplasmic space, I'm seeing the porins, and I'm seeing the outer membrane. So now let me start asking you questions from this. If I ask you where do you see the outer membrane, this table is done. Outer membrane compensation ke liye gram negative has. If I ask you where do you see the periplasmic space, you will say again gram negative has. If I ask you where do you see the porins, you will again say this is something that only the gram negative has. More or less it is done. Now I have to dwell deeper into this entire concept. So repeating all of this before I go deeper into it. For your gram positive, you will learn PPPT. The P is still pending and I'll tell you where. You will say it has a lot of peptidoglycan and it has teichoic acid for tikna and attachment. Rest everything else goes to gram negative. So if they ask you where is periplasmic space, where is outer membrane, where are porins, all of it is in the gram negative. Peptidoglycan nahi hai. Instead, it has lots of lipopolysaccharides. Before I go into the details now, I have to tell you guys what exactly is this lipopolysaccharide, because God has given it for a little bit of, importance. God wants that the gram negative organism should feel important. So God gave it a lot of lipopolysaccharides. You learn it as OPA. What is OPA? There are three regions that a lipopolysaccharide has. If you look carefully on the top, you have an O antigen. Guys, O antigen is the most variable part. Maximum variations in a gram negative organism are because of this O antigen. And I'll give you an example. You all have heard of a gram negative organism called Salmonella. Salmonella typhi. Basically, I'm talking about Salmonella typhi matlab I'm talking about enteric fever, typhoid fever. You must have heard in typhoid fever there is a test called the Vidal test, and in Vidal test, which antigen do we study? We study the O antigen. So there are two antigens we study, O antigen and H antigen. O antigen we have studied right now. It's in the lipopolysaccharide. H I'll come to in a while. So O I have understood. What is P? There is a little bit of polysaccharide. See, what did I say? Lipopolysaccharide. Na? So thoda polysaccharide it has. What is A finally? The lipo word, that means it is lipid A. So firstly I realize lipopolysaccharide mein lipid bhi hai, polysaccharide bhi hai, that I know. But there is O antigen also. So OPA, there is O antigen, then there is going to be lipo, the polysaccharide portion, and then there is going to be the lipid portion. So OPA, please yaad rakhna, from outside to inside. Antigen O, polysaccharide, and it is going to be the lipid A. Please note, you all must have heard toxins. You know, na, there's a very deadly list of toxins. You must have heard there's something called exotoxin, endotoxin. I'll tell you the difference. For the time being, note, this lipid A is going to produce or have the endotoxin. That is why you must have always heard endotoxin is present only in gram-negative organisms. Why have you heard this? Because endotoxin is present in this lipopolysaccharide portion, and that is only in the gram-negative organisms. And this endotoxin is known to have a lot of deadly effects. All the lethal fever wale effects come from endotoxins. So now note this lipid A. I have learned lipid A is very, very lethal. Lipid A is very, very lethal. Why? Because it has the lethal endotoxin residing inside it. So all your details of a lipopolysaccharide are done. Over here I'm going to write a chota sa OPA, because O antigen, polysaccharide, lipid A, lethal, endotoxin, story is done. Now I'm going to go into the details of the cell wall. I want to know their amino acids and all of that. So have a look at this. So first and foremost, I don't want you to read the table. I'm going to zoom into this picture and I'm going to show you what are the primary components. Anyone, chalo, let's zoom into this one. Okay, let's not go into too much of detail. I can see there are two structures. There is something called NAM and NAG. Please first do the full forms. What is NAM? N-acetyl-muramic acid, and NAG is N-acetyl-glucosamine. So I have realized a gram-positive cell wall also has NAM and NAG, and then I realized that a gram-negative cell wall also has NAM and NAG. So dono ke paas NAM, NAG hai. Okay, now I will obviously have to find what is the difference. Abhi difference nahi hai. Both of them have NAM and NAG, and I realized to this particular, let me trace. For example, this yellow wala is the NAM molecule, right? I can see that that's the NAM molecule. Do I see something attached to the NAM molecule? Yeah, I can trace it. There is 1, 2, 3, 4. 4 amino acids are attached to the NAM molecule. Let me go to the gram-negative organism. Yes, this is NAM, and 1, 2, 3, 4. 4 amino acids are attached to the NAM molecule. What are those amino acids? Yahan par difference aayega. So please note, for gram-positive organisms, I have learnt agla. What is agla? These are the 4 amino acids that are there, out of which A and A, you start with alanine, you end with alanine. Alanine, glutamine, lysine, alanine. Alanine, glutamine, lysine, alanine. Then again alanine, glutamine, lysine. So agla, that is going to be gram positive. In gram negative, you follow the same agla. But one of them is going to change. It is now going to become agma, because gram negative mein M wala alphabet aayega. Agma, that means A and A obviously remains the same, that is alanine, alanine. Glutamine also remains the same. This lysine is replaced by mesodiaminopimelic acid. So some M is gonna come. Mesodiaminopimelic acid is gonna come. So remember, gram positive mein what were the 4 amino acids? Agla. And gram negative mein it is agma. So right now also you will say, ma'am, 4 amino acids, same hi lag raha hai. Now tell me the difference. If you look at the difference, I don't know how many of you have noticed it, I see a horizontal connection also between these 4 amino acids and the next 4 amino acids, like these 4 over here and these 4 over here. I see a connection of 5. Can you count with me? 1, 2, 3, 4, 5. I only see it in gram positive. I don't see any connection in the gram negative. So now if I ask you 5 amino, 5 for what? Pentapeptide. Which of the cell walls has a pentapeptide cross bridge? And that was your last P that you had to study for gram-positive. The pentapeptide cross bridge is seen in gram-positive organisms, and that was the part of the table. See, I told you NAMNAG to dono mein hoga. NAMNAG is going to be present here also. NAMNAG is going to be present here also. Over here, along with this, there are 4 amino acids called AGLA, and in gram-negative, there are 4 amino acids called AGMA. That I know, but which of them is having a cross bridge? So remember, gram-positive has a pentapeptide cross bridge, whereas gram-negative does not. You can complete your PPT mnemonic. The last P was the pentapeptide cross bridge. Now, whenever you have to revise this, now that you've understood everything, whenever you have to revise, you're going to just learn PPPT. Pehle ye PPPT pad lo, then everything goes in gram-negative. So, in gram-positive organisms, there is peptidoglycan, there is pentapeptide cross bridge, there is teichoic acid. Right? So remember, peptidoglycan, pentapeptide, ye jitne bhi pemta, peptido, these things are there, whereas all the other P's are gram-negative, jo rare wale hain, periplasmic space, porins, lipopolysaccharides, outer membrane, all that goes in gram-negative. So let me take a test. Come on, what is this cell wall? Gram-positive, gram-negative. So you will say, ma'am, I can see NAM and NAG, wo to dono mein hota hai. I see something over here called AGLA. Agla was mentioned. I think it is gram-positive, but the best way of confirming is, do I see a cross bridge? Do I see that 1, 2, 3, 4, 5 thing happening over here? Yes, I see that. If I see a pentapeptide cross bridge, I know for a fact that this is a gram-positive. I hope you all have passed in this. This was your cell wall, but all this is said and done. How do you do the identification? For the identification of a cell wall, your mnemonic is registered medical practitioner doctor. Obviously, all of us are registered medical practitioners. So what all are you going to do? Just ratta. No one actually does half the things. So R is going to be reaction with antibody. It will have an antigen, you react it with antibody. M is going to be either mechanically you test it, or you do a micro dissection, mechanical rupture or micro dissection. You can also use plasma. You can test the cell wall with plasmolysis. None of these that I do. So remember, reaction with antibody, mechanical rupture and micro dissection and plasmolysis are ratta. What I do is doctor, that is differential staining. Differential staining means I do the gram stain to find out whether it is difference, whether it is gram positive or negative. So what I'm actually doing is doctor. Differential staining is what I do. This is done. Now all of you, right now, sometime back you were telling me, ma'am, you had promised you will tell us the difference between an exotoxin and an endotoxin, and we've never understood it. I know most students, even when I was in my MBBS and in my internship, I never understood this difference. So I think now we can. First, what is the basic that you have? Where did I teach you was an endotoxin present? Endotoxin was made in that OPA wala layer, the lipopolysaccharide. Lipopolysaccharide means that it is present in the gram-negative cell wall. So opposite karo, exotoxin is going to be present. It is not made up of lipopolysaccharide, it is made up of proteins, and it is present in both gram-negative as well as gram-positive cell wall. So opposite ho gaya. So my advice is learn one side, endotoxin yaad kar lete hain. Exotoxin we will be studying elsewhere. So now let's do a repeat. So please remember that endotoxin, which is a lipopolysaccharide, is a gram-negative organism and is present in gram-negative organisms. It is heat stable. It is number one, heat stable. It has low antigenicity. It is therefore needed in large amounts to create effects. Means that some molecules of endotoxin will not be able to create a problem. You need endotoxins in a good amount for them to create a problem, and whatever problem they will create, they will always create a constant problem. And how will they be released? They will be released on the lysis of the cell. So if I show you a particular picture, all of you can very well appreciate over here that if I enlarge into this, you'll say, oh ma'am, I can see the OPA, I can see the O antigen, I can see the polysaccharide, and I can see the lipid A. And you had told us that the lipid A is the part which is lethal, that is going to contain the endotoxin. And if I want this to be released, there will have to be lysis of the cell. So what all do I have? Once I want it to be released, I will have the lysis. Little amount is not enough. I will be needing it in a good amount. Large amounts are needed so that I can create an effect, and whatever effect I'll create, that will be a constant effect. And why is it needed in large amounts? Because it has a very low antigenicity. But yes, it is heat stable. Talking about exotoxin, everything opposite. Exotoxin is going to be released as secretion. So have a look at that picture again. This was probably a gram positive or a gram negative organism, because exotoxin can come from anything, and you can see it's these molecules. They are just secreted. Exotoxin will just exit, is how I have learned it. Exotoxin is just going to exit or is going to get secreted. It doesn't need any lysis or anything. It is just going to get secreted. And please note, little amounts are also going to produce a lot of effects. Why? Because it is very antigenic. It will just escape, exit karega, and it is going to create a lot of effects. And every time it is going to create variable effects. Every time it creates a variable amount of effect. Okay, so obviously once I've studied this, your main point is that, ma'am, whatever toxin you have, you should be able to make a vaccine out of it, hai na? So whenever I have to take a toxin and I want to make a vaccine out of it, I have to convert it into a toxoid. So which is the one in which I make a toxoid? I make it from an exotoxin. So for example, whenever you heard na, like कोई भी टॉक्सिन क्रिएट हुआ है, that toxin has made a vaccine that has been made from the exotoxin, because that you can convert into a toxoid and that can be used for your different vaccinations. Okay, now comes the story of exceptions. So what is the story of exceptions? I have told you that all the gram negative organisms will have an endotoxin. So mostly in the gram negative organisms, you're going to get endotoxin. Is there any gram positive organism that can show you endotoxin? Let's do a repeat. The only gram positive organism which shows you endotoxin is Listeria. So please remember this for your later part. Only and only Listeria is the gram positive organism that can show you endotoxin. Point number 1. Very important for you to know. Next, I told you that exotoxin hamesha exit karega. It's always going to get secreted. It's always going to exit. But there is an exception. Botulinum toxin is released on lysis. So please note, that's the other exception. Botulinum toxin, although it is an exotoxin, I expected that it should be exiting or secreted, but it is going to be released on lysis. These two are the most important exceptions. I told you, examiner is more interested in the exceptions. One thing I want to ask you from your pathology lectures, tell me something that you have already told me that endotoxin jo hoga, wo lysis hoga, and it's gonna come out and create impact on the body. So which is the toll-like receptor that it binds to? Can you tell me this question from pathology? Endotoxin binds to, and yes, in pathology I have taught you toll-like receptors. It binds to toll-like receptor 4. It binds to TLR4. So you will say, ma'am, is there any test that you can do in your microbio lab? Only for a quick review for INI-CET aspirants, ye jo animal hai, this is a horse crab, or looks like a horseshoe, right? This is the shape of a horseshoe. So this is a horseshoe crab, and that is known as Limulus. So what you do a Limulus amebicide lysate assay. It is known as LAL. I call it LALA. Why LALA? Because I call it Limulus amebicide lysate assay. What are you going to do? You are going to take this particular chemical from this animal. You take the Limulus amebicide lysate and you mix it with the sample, whatever sample you want to find out, kya, does this have endotoxin or not? If this becomes a little jelly-like, if this converts into a gel, this means that the endotoxin is present. So remember, if there is gelling of LALA, this means the endotoxin is present. That is how I remember. If there is gelling of LALA, this means the endotoxin is going to be present. What is LALA? Repeating, the Limulus amebicide lysate assay. Only as a point, this is important for INI-CET. What if cell wall pura ho gaya? What if the cell wall is not there? What are cell wall deficient forms known as? That is known as L forms. Firstly, why did I call it L? You can see a lot of L's over here. This was identified in the Lister Institute in London. So this was identified in the Lister Institute in London. The organism was used, that was Streptobacillus moniliformis, all the L's, and the scientist who discovered it was Klinberger. So I've got you all the L's together. L form was identified in Lister Institute in London. It was studied on Streptobacillus moniliformis, and it was identified by Klenberger. That means cell wall deficient forms are there. You know how have we divided it? There is something known as stable and unstable forms. Means what? Unstable cell form L forms and stable L forms. So you will say, ma'am, the first concept is L form. You told me that cell wall is deficient. So stable L form will mean that cell wall is deficient. Once it is gone, it is gone. It's never going to come back. Which is that organism that we have? Mycoplasma. Mycoplasma is not going to have a cell wall. Stable. Stable means it's decided, I'm not going to have a cell wall. It is never ever going to change its decision. It's never going to suddenly get a cell wall back. Once the cell wall is gone, it's gone. Stable. Headstrong. Okay? Stable L form means permanently cell wall is gone. That is mycoplasma. Unstable L forms are a little concerning because sometimes they lose their cell wall, sometimes they get it back. So it's a very unstable kind of an up-down happening. Firstly, you will ask me, why does something just lose its cell wall? Whenever it is treated with something like penicillin. So if you treat these organisms with penicillin, they will lose their cell wall, and after some time they might come back to the cell wall wala form. तो बहुत instability maintain हुआ है. These unstable cell forms, L forms, are of two types, protoplast and spheroplast. Remember, protoplast is going to be noted in protoplast, gram positive, and pura cell wall is lost. Pura means entire cell wall is lost. Did you get the mnemonic? When you're talking about a protoplast, it is seen in gram positive organisms, and pura cell wall is lost. Pura means whole cell wall is lost. So remember, protoplast positive, pura cell wall. So spheroplast becomes opposite, na? Spheroplast is going to be seen in gram negative, and is the pura cell wall lost? No, only a part of the cell wall is lost and a part of the cell wall remains. That's why I made this mnemonic, so that your life becomes easier, because till I was in my MBBS, my life was not that simple and we could never understand these things. So the purpose of me teaching microbiology has only come from the fact that I have cried over this subject once upon a time. Okay, so having said that, the protoplast gram positive में दिखेगा, पूरा cell wall is lost. Spheroplast gram negative में दिखेगा, little bit cell wall is lost. So repeating, unstable cell L forms, sometimes they can go here and there, with cell wall, without cell wall, instable है. And stable matlab mycoplasma, that is what is asked. So again, this is also done. Now let's move on to the third layer. So where are we, guys? We have done the outermost layer. The outermost layer was the capsule, or you can call it a slime layer, whatever you want to call it. Next, we have studied the cell wall. If it was a gram negative organism, I would have had an outer membrane also. Remember, I told you there are some extra layers that God has given it to compensate. So it is an outer membrane only if it was a gram negative organism. And finally, so I have nothing new to teach in that. Finally, I have something called the cell membrane. That is the next layer that I am going to talk about. So in this cell membrane, please note, you are put up a couple of questions. What did I say? Dekho, you did capsule, you did cell wall, you said maybe there will be an outer membrane also that is present, ho sakta hai, but I am now interested in this cell membrane. And can you see the cell membrane is dipping inside? Can you see this dip over here and this dip over here? This is known as a mesosome or chondroid. What is mesosome or chondroid? Basically, how does the bacteria breathe? What is the respiratory unit of a bacteria? That is what is a mesosome. So there are three questions of a mesosome. Question number 1, mesosome or chondroids are what? They are respiratory units. Question number 2, they are seen in both gram positive, gram negative, but they are prominent in positive. Remember, they are more prominent in gram positive organisms. And question 3, which is in front of you, it is an invagination of which layer? It is an invagination of the cell membrane. Quickly repeat, it serves as what? A respiratory unit, prominent in gram positive. Matlab you will see it in gram positive as well as gram negative, but more in gram positive. And it is going to be an invagination of the cell membrane. Now let's move forward. What are the other things that a bacteria has? It has ribosomes. They are 70S ribosomes. Thankfully, they have 70S. They don't have 80S. Why? 80S to the eukaryotes have, na? 70S because all the antibiotics can act on 70S. Hai, so 70S in a configuration of 50S and 30S. So that is the ribosomal story. If you ask me what is the kind of a nucleus that a bacteria has, nucleus that a bacteria has is single and circular. So if I have to draw a bacteria, inside a bacteria, I will always always draw a single circular, but remember it's going to be double stranded. It's a single circular double stranded DNA that this has. So definitely there is a nucleus present. Are there inclusions also? Some of the bacteria have inclusions, like example Corynebacterium diphtheria. I won't go into details, but for the time being those inclusions are known as volutin granules, right? Some energy reserves also some bacteria have. Volutin granules is a 20-minute topic which I have covered in Corynebacterium diphtheria. We don't have to repeat it here. So now if I go all the way back to my first slide, extra things that I have learned is, I'll label this entire thing now. So what is this? Ye jo respiratory unit tha ma'am, we have studied these are going to be the mesosomes. You have also taught us that this has ribosomes. Ribosomes are of what configuration? They are the 70S ribosomes. Then you have taught us that there is a circular double stranded nucleus also, DNA also that is present. Then you have told us some inclusions can also be present sometimes. Now it is time for you to tell us that is there any locomotion happening? Is there any flagella coming out? Yes, that's the next topic that we study. What is a flagella? The first thing I told you, flagella was for locomotion, was for movement, but that's not the only thing. That flagella also has an antigen called H antigen. So whenever you have a bacteria and a flagella is coming out, now we'll start drawing bacteria like this, and if a flagella is coming out, so of course it is going to help in movement, but it is also going to have the H antigen. Does this remind you of any test, guys? Sometime back I was telling you the Vidal test that you do for typhoid fever, and I said in Vidal test you measure two antigens, O antigen and H antigen. We've done both. O antigen was there in the cell wall, OPA wala cell wall means gram-negative cell wall, and H antigen now I know is present in the flagella. So this means this typhoid must be a locomotive, flagellae wala organism, right? So these are the two antigens that I test in Vidal. I know the meaning of both the antigens. Perfect. So this is an antigen. If you want to learn the dimensions, it is 3 to 20 microns long in terms of length. It is 3 to 20 microns. And in terms of the width, of course, it is very tiny, 0.01 to 0.013 microns. I don't know why have they even written thick, because that is not even the resolution of a light microscope. The resolution of a light microscope is also not 0.01 microns. I won't be able to see it on a light microscope. How will I see a flagella eventually? I will have to put some kind of a stain I will have to put. So I will come to that for the time being. But before, I think you know the stains. I have taught you a lot if you've read the bacterial section. But before that, all the flagella I was drawing, kya flagella is coming out. If I say there is a flagella but it's not coming out, अंदर flagella hai, endo flagella. So please remember, endoflagella is something that you see in spirochetes like those Treponema, Borrelia, Leptospira, spirochetes, and the origin is going to occur from the periplasmic space. Remember in gram-negative organisms, I told you there is a periplasmic space. That is the only question you get on a periplasmic space. That periplasmic space is going to give rise to that andar wala flagella, endoflagella, and that is going to be seen in spirochetes. So this is also done. Now let's go into the structure of a flagella. How does a flagella look like? Okay, I can see that there is a basal body over here. This much of the area that we have, that is going to be the basal body. Then I have something hooking out like this. So that happens to be the hook. And finally, the part that is free wala part is filament. So it has three parts. It has a basal body, then it has a hook, and then it has a filament, right? You can see that, everyone, that is arising from the cell wall. But you will ask me now that why have you written gram-negative organism? This means the flagella of a gram-negative and gram-positive bacteria is different. Yes. See, parts are the same. Basal body wahi hai, then there is a hook and a filament. Same way, basal body, then there is a hook, then a filament. That is not the difference. The difference is that the basal body has 1, 2, 3, 4 rings over here, and the basal body has only 2 rings over here. So remember, in a gram-negative organism, you have 4 rings, that is ring number, or you have named them MSPL. MSPL, there are 4 rings, whereas in a gram-positive organism, there is only going to be Miss. So I have learned Miss Preeti. Miss Preeti, you will actually see this, by the way. Get ready that in microbiology, I've been very selfish and a lot of mnemonics are on my own name because I was so struggling with them now, so I made mnemonics on my own name. That way you will remember me and you will remember micro. So remember that M and S are the only ones that you have in the gram positive, whereas all the others, that is P and L, all of those are going to go under gram negative. So MS in gram positive organism. So flagella is done. Now let's look at their location. If there is only one flagella like this, monotrichous, what do you mean by monotrichous? How have I learnt it? So one flagella on the side, and that is going to be known as mono. Write with me, monotrichous. Mono means one. That basically means that Vibrio sounds like one and pseudomono or pseudomonas. So pseudomono is also going to have one flagella on one side. So simple, Vibrio has one and pseudomono has one. Then we have something known as amphitrichous. Amphi matlab who are, like for example, you must have heard people who are ambidextrous. Ambidextrous are people who can use left and right both hand. So ye amphi, like I remember till almost my 8th grade, when I've been a left-handed person all my life, but till the 8th grade I was ambidextrous because I was using both my hands, and then obviously as studies increased I became left-handed purely. But even till date sometimes I can use. So the ambidextrous people are those who can use both their left and right hands as dominant hands. So amphi also means left and right dono hai. So you can see a flagella here and you can see a flagella here. What is lophotrichous? Lopho means there's a tuft. That is how I have learnt it. The word lopho is going to mean an entire tuft. So let's write it down. The way you say lophotrichous, you are going to see an entire tuft of flagella at one end, and the bacters show you that. Who are the bacters? Helicobacter, Campylobacter. So jitna bhi bacters hai, I'll write it for you. Helicobacter, Campylobacter. These are all going to be tuft or lophotrichous. Finally, coming to peritrichous. Peritrichous tells me it's all around the periphery. You will see the flagella everywhere. So when I write peritrichous, the most important ones that I have to know for peri, PR I will say for Proteus, and E I will say for E. coli. So peritrichous is going to be seen in Proteus, it is going to be seen in E. coli, and instead of ritrichous I will say Listeria. If you can learn, not very important. That's why I gave you the primary mnemonic that Proteus and E. coli are the most important. ठीक है, a quick revision. Monotrichous, mono matlab pseudomonos, and we have one that is Vibrio. Then we have lopho for a tuft, that is seen in Campylo and Helicobacters, and peri is seen in Proteus as well as E. coli. So on the basis of these locations, how many types of motility are known? Firstly, tumbling. Tumbling means like a drunk man. Tumbling motility is seen with Listeria. Listeria is your drunk man. Tumbling motility is seen with Listeria. In fact, when you go to Listeria chapter, it's an amazing story I've told you of a tully drunk man. Always remember, tumbling motility is seen with Listeria. Then comes darting. Darting basically means something like a shooting star. Something like a shooting star, like you can't even see, it's going so fast. And there is going to be two things. Number one is going to be Vibrio and the other one is Campylobacter. One of them is Vibrio, another is Campylobacter. Coming to the next, corkscrew, which is the organism that looks like a screw. Looks like a screw means, ma'am, it's looking very, very tight, like a screw, like a nail, like a screw. And that is going to be Treponema. Treponema. So we have always learned Treponema looks like a tight screw. Treponema looks like T for T, a tight screw, and that is why it shows you a corkscrew motility. Coming to twitching. Firstly, you will do Ratta. Twitching is seen with Eichinella. And twitching is also seen very famously with a T-wala organism called Trichomonas. Trichomonas also shows you the twitching motility. Trichomonas vaginalis, I hope you've heard about it. Trichomonas vaginalis causes strawberry cervix. Now that shows you T for T, twitching motility. Falling leaf. Who is the falling leaf? Girta hua leaf. Girta hua leaf is Girta for Giardia and leaf for Lamblia. Girta hua leaf is going to be Giardia Lamblia. Abhi tak ka repeat kar lo, because after this I'll tell you some advanced concepts. Tumbling is going to be seen in the tully Listeria. Darting is very, very fast shooting star seen in Vibrio and Campylobacter. Corkscrew, the one that looks like a tight screw, that is T for treponema. Twitching is primarily seen in Trichomonas, also in Echinella. Falling leaf, girta hua leaf is Giardia lamblia. Now I'll tell you what is differential motility. Means that if you have a lower temperature somewhere around 22 to 25 degree, it's going to be motile. However, if you have a higher temperature at 37 degree, it's going to be non-motile. So at one temperature it's motile and at the other it's not. And there are two organisms that come under it. There is something known as, I've learnt it as a rhyme, there is Yersiniosis and Listeriosis. So now I can say that Listeria was that tully man, Listeria was that tumbling, falling, drunk man. So that drunk man is going to be drunk at 22 degrees and is not going to be drunk at 37 degree, which is a life lesson that I'm telling you, that when you are young, you can still afford to be drunk, because at a young age your body will be able to accept that much of alcohol. But as you go in your 30s and mid 30s and beyond, your body will not accept that much of alcohol and you won't be able to get so drunk. So repeating once again, to tumbling and drunk at 22 years of age and not able to get drunk at 37 years of age. That is what is Listeria, the tumbling. Same phenomena also shown by Yersiniosis. Finally, I can see there is something called swarming written, and I'm not focusing on it. Why? Because many people say swarming is a type of motility. Some people say swarming is this concentric, it's a growth pattern. Rather than a motility, it's a growth pattern. So first and foremost, the picture that has been shown over here, the bacteria is having one circle, then it is making another circle, then it is making another circle. So the bacteria is swarming. Now many people believe that swarming is seen only with Proteus, but no. I have a mnemonic which is quite a lengthy mnemonic that all of you have to know. So we are now gonna pick it up. So where all do you see the presence of swarming? This will definitely help you in your exam. So when we talk about swarming, you have to learn the mnemonic PVCs. I'm finishing it off here so that your bacteriology section becomes easier. So P is going to stand for Proteus. VP together means you're talking about Vibrio. Which Vibrio? I told you VP na. So Vibrio parahemolyticus. There is another Vibrio that you have that is known as Vibrio alginolyticus. Vibrio alginolyticus is also going to show. Then C for Clostridium tetani. C for Clostridium tetani. There is one bacillus which starts with a C, and that is Bacillus cereus. Bacillus cereus. And finally, there's an organism with S called Serratia. Let's do a recap. What is swarming? Swarming is shown by PVCs. Swarming is going to be basically a kind of a blood agar, and on that blood agar, you're going to see a lot of these round movements. That is swarming. So P stands for Proteus, V stands for Vibrio parahemolyticus and alginolyticus, C for Clostridium tetani and Bacillus cereus, and S is going to be for Serratia. So coming back, this is what you have for swarming. It is going to show PVCs. That is your final flashcard for the types of motility. Now you need to know next, how are you going to demonstrate motility? So you have a couple of methods. One by one, I'll explain it in 5 minutes. There's something called a hanging drop motility. What is hanging drop? Have a look at this picture. I took a cover slip. I put one drop. Can you see? One drop of the bacteria can be seen, right? On top of that, so this is that cover slip that I have. On top of that, I've put a slide. And I can see that slide has this gaddha. It has a depression over here. So what am I doing? For example, if this is the cover slip, on this I have kept my organism, right? So I've kept my cover slip over here, and on top of this I am going to put that slide. I'm going to put that slide and then I will invert it. So this hand of mine is the cover slip. I have put a slide on top of it and I will invert it. So once I have inverted it, all of you can see, I have made this combination and I have inverted it over here. Will you see something hanging? From this cover slip, will you see something hanging? That is why it's called as the hanging drop motility. So just this, that you don't have to know the procedure, but you need to know, acha aisa kuch hua that you have a slide with a gaddha and something is hanging into it. That is hanging drop motility. Other than that, you can use a semi-solid agar. What is a semi-solid agar? Let me tell you, agar is usually used as a 2% concentration. Bacteria can't move a lot over here. But if I use a semi-solid agar, means I'm using it at a very less concentration, 0.5% concentration. It's a little softer, bacteria move karega. If it's softer, bacteria will move. The third one is Craigy's tube. Craigy's tube is in front of you. This is known as the Craigy's tube, and the other one that you have, which looks like a U, of course, is known as a U-tube. Okay, the U-tube. So now you don't have to know the pathogenesis again. You just have to listen to me once and then just know their name. Craigie tube and U-tube are for movement. Okay, so what do? You have a tube over here and you have kept another capillary inside it. So tube mein tube rakh diya. You are going to put your bacteria out here. Please listen to me carefully. You have decided to put the bacteria over here in this chota wala tube. This is where you inoculate the bacteria, and the bacteria is going to go all the way here. So you will never ever expect कि अच्छा छोटा वाला tube में था ना. Next morning you realize that you are able to isolate it from the बड़ा वाला tube. You are able to isolate it from the surface. You will say, oh, how has the bacteria reached here? It must have traveled. I left the bacteria over here, but the bacteria was motile. So it went from here and I could get it over here. So it has moved. That is Craigie's tube. What is U-tube? If I put the organism over here this time and next time I get it from here, so this means the organism has moved. You put it at one end, wo swimming karta it has come to the other end, and if you see it from here, that is the motile organism. Simple. If you put it in small tube and get it from big tube, or if you put it on one end and get it from the other end. These are the principles of a Craigie's and U-tube, and that means that your organism is motile. Again telling you, these are not need to be known in detail, but you've understood the concept once. If these are available, you do this, but if you have to look at it under the microscope, now you will definitely tell me that can you look at it under the light microscope? Usually, no, I can't. Why can't I look at it under the normal light microscope? Because it's very, very thin. Remember, 0.01 microns to nahi dikhega. So firstly, I can use a stain or I can use a dark field microscope. So one possibility is that I start using a different microscope. That is a dark field microscope. If you don't have a dark field microscope, you go in for a stain, and the name of the stain is the Leifson and Reuss stain. The name of the stain is the Leifson and Reuss stain, which is very, very important. You don't get the entire pathogenesis of it, but please note that the mordant used over here is tannic acid. The mordant used over here is going to be tannic acid. So you just have to know that for the flagella, we are going to use the Leifson stain. Flagella are done. So if I draw this as the bacteria, one of the projections was a flagella. There's another tiny little projection, and that is known as the fimbria. So we've defined it. It's very, very tiny. Pili matlab like hair. So whatever you'll see as the bigger organism, the bigger thing coming out, that will always be the flagella. And the tinier one that you'll see coming out, those will be the fimbria, because they look just like hair. And this fimbria is also known as pili. They are also known as pili. So first you will ask me, बस ये show off तो नहीं है? No, these are not show off. They also have an antigen. So I realized flagella also had an antigen. What was the name of that antigen? H antigen. Fimbria also has an antigen. The only difference is that it is a little smaller like hair. What is the primary purpose of a fimbria? Like, tell me, what was the primary purpose of a flagella? Flagella was for movement. What is the primary purpose of a fimbria? That is attachment. So when it goes, and you will see that, okay, it's attaching onto a cell. It comes in our body and attaches onto a cell. That is what it does by pili. But if this attachment, if this pili is used between a male and a female for mating, if it is consumed for sex pili, that process is known as conjugation. So for example, if a male bacteria and a female bacteria want to combine, they will combine with the help of pili, because it's for attachment, and this process is known as conjugation. So how will I find out whether there is a pili that is present in a bacteria or not? So you will say there are two tests for the same. Firstly, we have something known as heme agglutination. These are just tests which no one does, but again, for your exam. For heme agglutination, if you do heme agglutination, you will be able to find out the pili. In fact, when you go to organisms like gonococcus, you will see that on the basis of the heme agglutination test, an entire classification is there. Next, you have to do a surface pellicle test. It is found that if you take a broth or a culture media, and if an organism has a pili, you will see a thin layer at the surface. You will see a pellicle at the surface. Basically, if you see a pellicle at the surface, this means that there is a pili present. So simple, pellicle hai, to pili hai. Pellicle hai, so this means pili is there. So now I want to ask you something. That means for all practical purposes that you are saying, flagella will always be for movement and pili will always be for attachment. Yahi padha? Okay, whenever I will teach you this, I will always teach you exceptions. So now I'm gonna make that thing that I told you right now. Flagella is mainly for locomotion and pili is going to be for attachment. However, can there be some pili called as type 4 pili, which is important in Neisseria? In Neisseria, type 4 pili is also used for locomotion. So you will suddenly realize that, ma'am, you are saying that Neisseria is motile. Yes, but it doesn't have a flagella. No, it doesn't. Why is it motile? Because this type 4 pili is making it motile. Okay? So the type 4 pili is making it motile, and that is what you need to know. It's a flagella independent motility that Neisseria shows you. Finally, coming to the last part of the anatomy, and that is the spores. Spores means slept off. The bacteria has slept off. So whenever there are any unfavorable conditions, the bacteria feels that I won't be able to survive. Let me sleep off. Let me become tough and resistant. So let me go into a resting or a dormant state. Right? Whenever there are any unfavorable conditions, the bacteria will say, let me sleep off. So first, you should know what are the different types of spores, and before that, know that there are two spore forming organisms that we have in the body. There is something known as, in the microbiology section, all the bacilli and all the clostridium are going to be spore forming. I'll give you a mnemonic. If you look at these, you will realize, let's start from here. There is one type of spore which is within the bacteria. Bacteria ke andar, it is not bulging out. One type of spore is creating a bulge like this. It is a bulging spore. So you will ask me, out of Clostridium and Bacillus, which is bulging, which is non-bulging? Please note, B is not for B. Bacillus, you can't put bulging over here. You will put the other one. This is non-bulging. This is non-bulging. And Clostridium, here you can put the B. So this is going to be bulging. So B is not for B. Bacillus is not going to be bulging. Clostridium is going to be bulging. So one end of it is done. Next, you will ask me the location. So if you look carefully, either they are right in the center of the bacteria, or they are right at the terminal of the bacteria, or they are just before the terminal. So please note, most common location is going to be subterminal. Most common location is going to be subterminal. However, you will tell me that where all do I see terminal spores? And this is a very easy way of learning it. TE for terminal. TE for terminal means Clostridium TE for tetni, TE for tetni, and Clostridium TE for tertium. Simple, guys. If you see TE terminal written in the exam, either pick up TE tetni or pick up TE tertium. Please note, tetni may, if I draw it, if this is the bacteria and you're saying terminal, I have made a round terminal spore. I have made a round terminal spore. Whereas in tertium, again I have made terminal, but I have made a very oval wala spore. I have made an oval terminal spore. What am I trying to show you over here? The other column, when is it round and when is it oval? Because that will tell me that tetni is known to have a drumstick appearance. You can see now it's totally round, so it's looking like a drumstick. Whereas tertium is going to have a tennis racket appearance. And how do I learn it? See, it's looking like a tennis racket, long like a tennis racket. So confusion hota hai, students get confused which is drumstick, which is tennis racket. So I will say that the one that is going to have TER, dekho, TER for tennis racket. TER is going to be for tennis racket. So now you will always remember that tertium was tennis racket and tetni was drumstick. Now finally, where do you see the central spores? In which case do you see the central spores? So again, you have to remember C for central, C for serious. C for central, CE for central, CE for serious. And Clostridium bifermentans. What is this? Bifermentans, because that tells me if something is in the center, it will divide the bacteria into two parts. Bi, it has divided it into exactly two parts, matlab it must be in the center. So repeating, all the spores are subterminal, but terminal mein TE tetni, TE tertium, central mein CE central, CE serious, and we have bifermentans. The spores are done? Not really. You need to know the parts of a spore. So if you look at the, let's start from inside to outside. If you look at the centermost part of a spore, that is known as the core. Anything that is in the center is obviously the core, and that is made up of dipicolinic acid. Please note that the core, so I'll write them out one by one. The core is made up of dipicolinic acid, and this is what is going to make it heat resistant. Dipicolinic acid is making it very, very heat resistant. That is why I said it can survive a lot of bad conditions. In unfavorable conditions, a bacteria makes a spore so that it can survive all these. It's become resistant to heat. So the first thing from the inside is the core. What do you see outside the core? Now you start going into the cortex. The next thing that you have, the second thing that you have over there is going to be the cortex. Fine. What do you have around it? Now it's time to make it wear a coat. Make it wear a coat. So the third thing that you have is a coat, and coat is going to make it chemical resistant. Again, another feature that can help it fight. Coat is going to make it chemical resistant. And finally I go on to the outermost, and that is known as exosporium. Exo means सबसे out, exit. Exo means it's on the outermost, that is exosporium. So two things are sorted. Center में तो हमेशा core होगा, of course. Periphery में exit, exosporium होगा, of course. You just need to know cortex and coat. And if they ask you which is inside and which is outside, तो coat तो you will always wear from outside, ना. तो coat is gonna be the outer one, cortex is gonna be the inner one. If you ever have to arrange them from inside to outside, so repeating once again, inside is the core, outside is the exosporium. You're left with cortex and coat. Coat को थोड़ा बाहर रखना. Now you will say, how did this form? Now this information that I'm telling you is only important for the second prof students and maybe for INICT, not really for NEET PG. So how is the spore formation going to occur? So for example, if I look at this bacteria, the bacteria was dividing apna badhiya se, but if there was any unfavorable condition, if there was any unfavorable conditions, the bacteria said, I will have to make a spore now. How should I make a spore? I can see starting from stage 1, I go all the way to stage 7. So from 1, you will start making the spore, and on stage 7, you will release the spore. So 1, 2, 3, 4, 5, 6, 7. On what stage do you release the spore? You release it on stage 7. There are 7 stages of spore. You don't have to know each one in detail. I'm telling you the ones that you have to know. So first and foremost, of course, when do you start it? There is in the starting, there is just a filament that is formed. So what is it? Tell me guys, when I say stage 1, and after this I'm just explaining it, after this I'll write it all down. When you're starting with stage 1, the first alphabet is A, so you're going to make an axial filament. After that you come to stage 2, you will say, ma'am, in stage 2 I can see the bacteria has only split off into 2 parts. One part is making the spore and the other part is normal wala bacteria. Bilkul, in stage 2 you have two parts. In stage 2 you have two parts. Okay, finally coming to stage 3. Whatever that spore you had made, bacteria says, now I will eat it. Stage 3, the way you write 3 ka ulta karo E, you will engulf the spore. You have eaten up the spore. Now the rest of the stages you don't have to know, because now in stage 4 you will make what layer? Cortex. Then thoda bahar wala you will make the coat. Then you will make all the maturity in stage 6, and in 7 you will release it out. Now I don't want you to learn all the others. So I want you to learn stage 1, 2, 3. After that in 4, 5, 6 you will just keep on making it mature, and in stage 7 you will release it. So let's do a recap. Let's do a recap of this once again. Stage 1 is going to be A alphabet, axial filament formation. Stage 2, you have two parts. One is the spore wala part, one is the bacteria wala part. In stage 3, you will invert the 3, you will get engulfment. You are going to engulf the spore. In stage 4, 5 and 6, you are just going to mature it. And in stage 7, you will, 1, 2, 3, 4, 5, 6, 7, you will release it. Is this okay with everyone? Because now we come down to almost the end. What are the stains for a spore? I have taught you this way back in the previous chapter, the starting of microbiology, that we can use something called the Zn stain. If we are using Zn stain, we will use a 0.25% of H2SO4 that we have, right? 0.25% of H2SO4. We can use another stain called the Schaffer and Fulton stain, which is known as the modified Ashby stain. And this is the picture. I'm not repeating it. I hope you remember, guys. I had said Miss Spore. And I gave you a correlation also. Remember, what is spore? Spore is the sleeping form. When you go to sleep in your house, if you're sleeping at odd hours like 2 o'clock in the afternoon or something, your family will always come and taunt you. Your mom or your dad will come and taunt you that, oh, this person is a prince or a king or a princess and he or she is sleeping till 2 in the afternoon. So they will always address to you with some kind of a taunting name, like Miss Preeti is sleeping at 2 in the afternoon, what a life. That is how they'll say. So Miss Spore is nicely sleeping. That is why I'm taunting it. Kya re, this is sleeping, let me taunt it. What is that Miss? Because this stain has malachite green and saffronin red. These are the two colors. There is a malachite green and saffronin red. One green, one red. Tell me, what is the color of the spores? Can we all see that? Yes, we can see the beautiful green color spores. That is a previous year question. That what is the color of the spore? What stain do we use? So spore ke liye we use S wala stain. Spores ke liye we use the Schaffer and Fulton stain, and it is going to be green in color, malachite green. So remember Miss Spore over here. There's another stain that you can learn, and that is known as the Moller stain. Again, something if you are interested in knowing. Not very important. Schaffer and Fulton or modified Ashby and Zn are the two main ones. Not right now, but in a while I will also tell you that spores are very important, because spores are quite tough. They are the resting forms. They are quite tough. They have formed in unfavorable conditions. So I will use them for the controls. Like the spores of Geobacillus sterothermophilus, means they can survive very high temperatures. I will use them as a control for autoclave to see if my autoclave in the department is working well. Similarly, spores of Bacillus subtilis, Atrophius or Clostridium tetani I will be using as a control in a hot air oven. Even if you're not getting this concept right now, never mind, the next chapter is going to be sterilization, disinfection, you'll understand. The only thing is that yes, spores are used as a control for a lot of sterilization, disinfection techniques. Now, if you have studied all of the bacterial anatomy, before I end, I'm going to teach you a table which I ideally should have taught you in the beginning. But I never do that. I have always changed the sequence because I believe that now you won't have to mug up that table. And that is the table of difference between a prokaryote and a eukaryote. I know prokaryotes are bacteria. Now you've read everything about a bacterial anatomy. So you'll be able to make this column so easily, and opposite kar doge, you'll make the eukaryote. So let me ask you, did you have a proper nucleus? You'll say, no ma'am, there was just some weird single circular double-stranded DNA. That is what you had made for us. We had a very weird single-stranded DNA like this, right? So over here, we'll first see this. We see a very weird single-stranded DNA. This means there is no proper nucleus. We don't have a proper nuclear membrane or nucleolus or histones. ये सब जैसे humans में होता है वैसा नहीं है. Over here, we call it a nucleoid because all that it has is a single circular double-stranded DNA. Versus if I compare it with eukaryotes, yes, eukaryotes have all the proper, like all of us, we have a proper nucleus, and chromosome is going to be, in these there are multiple linear chromosomes that you notice. Okay, then tell me, in a while you will study bacterial genetics. Does the bacteria show you plasmids? Yes, bacteria shows you some extra chromosomal DNA also known as plasmids, whereas the eukaryotes show you mitochondrial DNA. The extra DNA is mitochondrial, like we also have it. Okay, did you see any organelles? Did I show you over here any other mitochondria or endoplasmic reticulum? No ma'am, you did not show me any of it. The only thing you showed me was a ribosome, and that was 70S. This means a eukaryote will have organelles and the ribosome will now be 80S, and that is what I told you na, that it's good that the bacteria has a 70S ribosome because all your antibiotics can act on this 70S. Now tell me, for the cell wall, did I teach you that the cell wall had N-acetyl muramic acid, N-acetyl glucosamine, all that was present na? But in the eukaryotes it's not gonna be there. Similarly, the cell membrane is gonna have sterols. Did I teach you that? No, ma'am, you didn't say. All that you told me was the cell membrane was gonna have that dip, wo respiratory unit wala dip. You did not tell me any sterol, because later on in a eukaryote a sterol will be present. Finally, where was the respiration occurring? That mesosome, that dip right now which I told you, mesosomes are present, that is where respiration occurs, but in a eukaryote, there are no mesosomes. So I realized whatever you studied for a prokaryote right now was bacteria ka story, and opposite becomes a eukaryote ka story. But there are some exceptions. Is there a prokaryote which does not have a cell wall? Of course, I told you some cell wall deficient form. I told you mycoplasma. I told you, mycoplasma, that does not have a cell wall. So when God has not given it a cell wall, God has given it sterols in the cell membrane, which is the prokaryote, which can have sterols. So God takes away one thing, he gives one thing. So if he's taken the cell wall away, he has given sterols in the cell membrane. Last one, I just told you that N-acetyl muramic acid, N-acetyl glucosamine, all these are present. Na? So is there any bacteria which lacks the muramic acid? And that is going to be chlamydia. That is going to be chlamydia. Again, these are important points on which I have wrapped up my entire bacterial anatomy, and we are ready for some questions. While examining a bacterial slide under the microscope, you notice that the organism lacks a cell wall. The cell wall deficient organism that could have been there on the slide. So which of these does not have a cell wall? We've just now studied it, that is mycoplasma. Let's pick up one more question. This is interesting. There is a sputum sample that was sent for testing in the microbiology lab. The gram staining showed presence of gram-positive cocci, which correlates with the cell wall properties. Which of the following statement is false regarding the cell wall? देखो कौन सा cell wall है? We are dealing with gram-positive. You probably have to think of that PPPPT mnemonic, and you have to find out the false statement. Let's do true-false. Is the cell wall of a gram-positive thicker? Yeah, it has more peptidoglycan. P for peptidoglycan. Of course, it is thicker. Correct. Look at the next one. Teichoic acid is present in the cell wall of gram-negative. No, I just told you PPPT. Teichoic acid is present in the cell wall of gram-positive. This is the false statement. I found my answer. But still, I'll read all. Region 2 of a lipopolysaccharide is core polysaccharide. Yes, I learned lipopolysaccharide was OPA. OPA means there is O antigen. Lipopolysaccharide ka P was polysaccharide and A is lipidase. So is region 1, 2, is region 2 a polysaccharide? Yes, this is also true. Lipopolysaccharide is responsible for O antigen specificity in gram-negative bacteria. Yes, it has an O antigen which shows you variability. So of course that is also true. So the only false statement over here was teichoic acid, because teichoic acid is not present in a gram-negative, it is present in a gram-positive organism. We finished off the entire detailed bacterial anatomy, and now we are going to move on to the next topic, and that is going to be the bacterial shapes and the bacterial physiology.